Hello everyone, welcome to today's webinar. I'm Kelly McSweeney, Contributing Editor at CNEN Media, and I will be moderating today's event. This webinar is titled Solid State Chemistry, Optimal Form Selection by Controlled Crystallization, and is being sponsored by Mercachem Syncom. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by Mercachem Syncom. Mercachem Syncom is the leading mid-sized European drug discovery contract research organization for your chemistry challenges. With over 450 scientists, they offer best-in-class chemistry solutions for drug discovery and drug development, from small to medium-sized molecule hits. Their services span across early-stage hit finding all the way to the delivery of your early clinical drug substance API. They blend vast scientific knowledge with inventiveness to make great leaps on your behalf. During the presentation, we will hear from Dr. Edwin Aret. Dr. Aret holds a PhD in solid-state chemistry from Radboud University, Nijmegen, the Netherlands. He has nearly 25 years of experience supporting every facet of the pharmaceutical development cycle, from discovery to commercialization, covering small molecules, drug substance, and drug product. Edwin's in-depth expertise includes crystallization, form selection, stability studies, solubility determination, bioavailability, and crystal habit optimization. Edwin is currently the principal scientist for solid-state chemistry. I will now hand it over to Dr. Aret.
Thank you for the kind introduction. The Solid State Department of Mercachem Syncom provides for already 10 years now single physichem analytics, standalone form screens, and also fully integrated early phase development programs on solid states together with chemical development as well as analytical development. Solid state is a broad expertise, combined analytics and experiments to support the pre-formulation R&D as well as GMP manufacturing. Today, I will talk about crystallization and form selection. The most asked question is when to do a solid state program. Well, I always advise not to wait until there is an issue with the form and you need to repeat any of the studies. In the initial phase of discovery, there is the characterization of the forms that you find. While in preclinical phases, there is the selection of the right salt or polymorphic form of the material. While when you are in clinical phases, there are the patent screens to provide the IP protection of your compound. So what is actually crystallization? Well, that is very much depending on who asks. The scientist will say, well, it's self-assembly of molecules to form a highly organized crystal lattice. That's wonderful, but that doesn't give you a better compound or a good product. When you ask process operator, it says, well, this is just a separation technique. We want to isolate the liquids from the solids. And the analysts say it's a way to purify the material. And when you look at from a solid state part, it is actually looking at different properties and you want to improve them by looking at amorphous versus your crystalline form, the selection of the right polymorph, improved crystallinity. In the end, all these forms will provide IP protection of the molecule. So here is an example. To the left we have some solidified oil, and that was obtained by Crest isolation. To the right there is the controlled crystallization that was done in a slow cooling experiment. It is exactly the same material, with the same chemical properties, it's only totally different in your solubility as well as stability and behavior. Crystallization consists of two different steps. First nucleation, next crystal growth. For nucleation, it only occurs when there is a driving force. So a supersaturated solution will be formed, and that only occurs when there is a higher concentration present compared to the equilibrium concentration. So the excess of compound in solution will solidify at a rate that is related to this supersaturation. Control of the concentration, as can be done by lowering the temperature, partly evaporation of the solvent, or adding an anti-solvent, will result in a driving force for nucleation. For crystal growth, the binding energy, which is related to the interaction with the solvent used, as well as the flux, which is controlled by the mixing of the solvent solution, are the rate determining factors there. Finally, during isolation, which is done by filtration followed by drying, no changes in your polymorphic form, solvated form, the crystallinity, and even the particle size and shape should occur. Polymorphism is the result, the kinetically first accessible form to be preferred to crystallize above the thermodynamically most stable one. So kinetically says what can you make, thermodynamically is what you want to have. Otherwise also the pharmaceutically acceptable forms, looking at an anhydrate or hydrated form, as well as a selection of suitable counterions or coformers. All is controlled by the nucleation, and the final properties of your material are decided by the crystal growth. Each phase has its preferred methods and solvents, a way how to work with the material, while in discovery the aim is to have just a crystalline product, and in preclinical phases it's selection of the most suitable form. So for discovery it is okay to have simple thermal cycling in process solvents, where your selection of the solvent is based on the solvent class or the active group. In preclinical, you want to have a diverse set of crystallization methods, as well as a broad solvent selection. And here it's more based on principal component analysis. Here you want to have the highest diversity, but still scalable methods. And crystallization is all about control. So a controlled nucleation, where you know the method, the solvent, and whether you have to use seeding. You control your crystal growth by a certain cooling rate, by an anti-solvent addition, and the amount of steering that you apply. It gives certain results. And these results are analyzed either by looking in situ by using probes, stability, amount, or by sampling, and you can analyze the material afterwards. The crystallization systems we use for the initial experiments, as well as for the optimization of a selected form, are the Technobis parallel reactor equipment. We have a crystal breeder, crystal 16, and a crystalline. The only difference here is the size, the working volume, and the scale-up steps are about 5 times that volume. All of them are parallel reactor systems, and they are applied with steering, heating, cooling, and turbidity. For the larger system, there is also video and the possibility to measure Raman. Then there is the question, is seeding actually required? Is it preferred or even wanted in a process? The control of the polymorph as well as the particle size distribution, that's the preferred part. But you do mix different batches together, which for GMP reasons can be less wanted. In this case, we have an example. We have a process R&D material, and actually there is only one suitable solvent. It is obtained by cooling crystallization, and the control is very poor. The bulk properties of this material, as you can see, are most dictated by the fact that these are combined fine needles and not single crystals. From the crystallization study, we got more understanding on how actually the nucleation part of this crystallization worked. It was mainly an oiling out of the compound, and finally it solidifies and recrystallizes in the form of needles. So really control about the nucleation part was not present in this crystallization. In this case, seeding gave more control about the crystal structure as well as about the particle shape. The formed material showed clearly single-sized particles that had a good filtration and drying rate, that had good properties, bulk properties, good flowability, as well as stability properties. In this case, seeding was required. Then the question about what is actually the best form. This is an example in which different companies and different techniques were applied to come to the answer, what is the best form. Here, first solid state was done. And there, the good viscous properties were selected. The material looks like small fines and it had a very fast dissolution rate. Next, R&D chemist was done. And the form obtained here, good filtration and drying properties, because these are large crystals. Totally different bulk behavior compared to what was selected by the solid state. Still exactly the same molecule, the same material. Regretfully, this was the result of the formulation. A hydrate was formed. These have very poor solubility, so it crashes actually out of the formulated product, and elongated, very thin needles were formed. Again, a different form compared to the previous two, and something that was really not preferred, as it has very poor solubility and solvent stability. So what was necessary was actually to repeat the form selection study to come with a new, better stable and better formulatable form. So form selection. Form selection is actually based on your material properties. So you can select a certain polymorph. That one will have a certain solubility, a dissolution rate, stability, bulk properties, equivalent to tablet. Selection of that form, most of the time based on stability. So we select the most stable form. You have to take into account that the most stable form is also the less soluble form. In polymorph studies, there is always a new form to be found. So the number of polymorphs is actually related to the time and the budget that you spend to it. When you look at hydrates and solvates, these are different unit cell, so it's not a polymorph. It is a hydrate or a solvate. It consists of different parts in the unit cell. Selection of a stable hydrate might be preferred above one of the most stable anhydrous forms. Because we have to take into account that the hydrate will be less soluble in water than the anhydrated form. So if it's being formulated in an aqueous buffer, you will get the possibility that the hydrate is being formed. And if that one is less stable or less soluble than the anhydrated form, there we get crystallization of your hydrate instead of a solution of the anhydrated form. Selection of a salt or a cocrystal is mostly used either for purification or to improve your solubility. In the case of cocrystals, most of the time it is improvement of the dissolution rate. Next, when you look at the crystal habit, there are differences in your crystal size and crystal shape, independent if it is the same polymorphic form or a different form. That will give you a different filtration rate, it will give different drying properties, different flowability, and in the end also different dissolution rate. So to improve your dissolution rate, you can go to a smaller particle by micronization. Or to improve the drying properties, you can go to a larger crystal, a less surface, and therefore it will be faster dry. Depending on the properties of the material, certain text needs to be adjusted and optimized. Looking at polymorphs, these are the same molecule with a different crystal structure. And they have different properties, either in activity, thermodynamic, melting point, solubility, stability, and looks different as a crystal, but each of these polymorphs can be patented. Here a clear example of different polymorphs. Needles to the left, block-shaped crystals to the right. The needles will likely have a good dissolution rate, but horrible drying properties, while the block-shaped crystals will have a good filtration, improved flowability, but yet it might be less soluble. So, which properties are important? For solid state, we look at solubility, stability, permeability. Optimizing the combination of the three, so balancing actually these properties, that will give you a good product. It's all about the strategy that will follow. Poor soluble form will require a different approach than a poor stable form. It is lowering the risks during your development and avoiding to have to go back in your research and have to select a different form as the first one was not optimal. So looking at these properties, when you go for solubility, you can select another polymorph. You can go for a hydrate, you can lower the particle size, or you can go for a certain salt or cocrystal. In case of permeability, that is most related to the molecule. So you can change your molecule, you can work with the carrier, or go for a prodrug. Stability, looking at crystallinity, and most of the time also particle size, amorphous content. To lower hygroscopicity, you can select a certain different counter-ion that on itself is not hygroscopic. And the selection of the less hygroscopic material is also very important in this case. This is the known ranking by the biopharmaceutical classification system. And what you see is, looking at the percentages, most of the solvent of the certain products that we get are the ones with the low solubility. So most of the work that we need to do in solid state, see how can we improve solubility in such a way that it will be going towards the class 1 or class 3. This is the overview selection that we do that is based on high throughput solid form screening. The aim is to have a broad overview. We do a lot of different experiments and you actually will have little knowledge of each individual experiment. The automation here is applied in solid dosing, in the crystallization step, as well as in the solubility determination. Sample handling is automated for the solids, measuring by XRPD, as well as by the liquids, measured by HPLC. The characterization is completed by standalone analytics, and all of the data is stored in a searchable and accessible project database. Solid form screening makes use of Unchained Lab systems. These are flexible platforms with separate components. These are all connected by the software. So we have a platform for solid dosing, one for liquid dosing, and one for crystallization. It aims for about 800 parallel experiments at the scale of well plates. Take into account that the high throughput screening results are only the first step. It is not the conclusion. The next step is to scale up and do a full characterization. So, which crystallization method should you use? There's a very broad set of different methods available, and these are either research-based, so the ones that are actually mild conditions and take very long to get you your solids, but there is also the production part, and these are actually based on fast isolation of your material. The advice is to restrict only to scalable methods within reasonable time. Find a combination of the research and the production-based. This selection is actually done to have a scalable form, but it does not limit you in the possibilities of different materials that you can get, different polymorphs that you can grow. Your selection of the solvent or your solvent mixture, that one determines your solubility curve. That one is fixed. But your nucleation curve, so the form you actually get by nucleation, that one is determined by your crystallization conditions. So the cooling rate, the anti-solvent addition rate, and evaporation rate. In all steps, the solvent selection is key. A limited diversity will only show a small selection of the possible forms that can grow. For your selection that is based on a solvent structure or the active group of the solvents, that results in about 20 different classes. The identification of the solvents based on principal component approach gave three most important properties: polarity, ability to get polarized, and the hydrogen bonding. Combination of these three properties gives you 8 different solvents at corner points and optionally one center point, as indicated in the figure to the right. Based on these results, you can actually predict the behavior of the solubility and the crystallization in the entire solvent space, as well as you are able to select similar but alternative solvents around a certain form. When going for a single form selection, it requires only a few solvents with the highest principal component diversity, and you can predict and have the overview of how they are related. In a pattern screen, you should use most different solvents. Here is a simple example of a polymorph study. You can find these in literature. These are different methods, and it's actually one scheme that fits all issues. You have different methods available: cooling crystallization, anti-solvent addition, evaporative crystallization. Use a slurry plate for recrystallization, an LC plate for determining how much is still in solution, or do we have decomposition. This looks very simple, and it's a nice base to start from, but it is not reality. In fact, when you want to start your study, you need to consider different concentrations. The control of your concentration is actually the point that will give you different nucleation, different forms there. In case you want to do a hot filtration for a clear solution, you can decide to go for different temperatures. When you look at a slurry crystallization or recrystallization, you should start with that form that is the least stable, or preferably the amorphous form. Slurry plate can also be done at different temperatures, as stability of the form you get can be temperature dependent. In case of a cooling crystallization, in the lab you can have about any rate that is possible there to test. But in production, you are limited to the possible cooling rate of the reactor, which is most of the time much slower than you can achieve in a well plate. So looking at crash crystallization compared to slow cooling will give you the overview if you have a different form depending on your cooling rate. In case of an anti-solvent addition, you can use forward or reversed addition. Addition rate can be changed as well as the amount of anti-solvent that you add. In evaporative crystallization, you should consider a vapor diffusion as well. This is not scalable, but it will show you in certain cases a different form that is available. And it will give you indication how do you do your drying. Quickly remove everything under high vacuum, use a nitrogen feed to your material to improve drying, or just leave it and let it crystallize in the vapor that is actually there. On your HPLC plate, you can also apply the different temperatures. You have your master plate at a certain temperature where everything is dissolved, and you have a crystallization plate at low temperature. The difference in solubility can be detected by just taking a sample for your HPLC at 2 temperatures. Of course, connected to high throughput screening are the analytics, and the bottleneck in the whole screen should never be your sample handling or the measurement time or your data analysis. So, next to your high throughput screening, you also need high throughput analytics. We have built an XRPD system that can do high resolution and high throughput in 24 hours to measure a full screen. HPLC solubility and purity is being measured in the same time frame, and both of these do not require new sample handling. The well plate is used as sample holder for the X-ray, and the dilutions for your LC are taken on the crystallization platform. This will give you a good overview of the properties of the material as well as the conditions that are required to get a certain form. Next question is, well, we have done the high throughput screening, so which polymorph to select? Just remember that the design of the study determines the outcome. So a limited diversity and also using the most stable form already as a starting product, that will give only a few different results. The most occurring form is not necessarily the most stable polymorph. In this case, it's the master plate, the different conditions, and we have an overview of the X-ray patterns found. This design resulted in 4 different crystalline forms, and it was clear that form 3 and form 4, these are solvent dependent. Only in one pure solvent these were obtained. But by looking at the results of form 1 and form 2, it shows to be completely random. Well, just a screenshot of the forms, that is not a conclusion. It is the dataset for the next steps. So each form found should be characterized from the well plate by looking TTA, is the solvate, a hydrate, or an anhydrous form. Do you see what are the thermal events? So can we already rank the different forms based on your melting point? Next step is to reproduce the experiment at about 200 milligram scale. You have sufficient material for further characterization, but you can also see if you have different forms that are very similar and you do not exactly understand how they are formed, what are the conditions to form that one, you can see how are they related to each other, what is the mutual stability or your solubility. And of course you can measure the interconvergence conditions. In the graph, you see the form 1 and the form 2 from this example, where the form 2 shows to be less soluble and therefore more stable at low temperatures. Your form 1 is more stable at higher temperatures. Transition temperature is around 55 degrees. So there are conditions in the screen that allowed form 1 to be the most stable form to grow. Certain cooling conditions go to form 2, and the mixture is also obtained. It didn't have enough time to convert into one form. When we did the test for the interconversion conditions, it was a slow process to go from form 1 to 2 or the other way around. Finally, once you have a preferred form, that one has to be reproduced at about a gram scale. You can have producibility, you can have a look at the crystal habit. At that scale, it starts to show actually the particle size and the particle shape that is also obtained at the larger scale manufacturing. Here is shown an example of the form conversion conditions. It allows you to repair a batch that has an undesired polymorph or mixture, also determines storage and handling conditions of one selected form. Knowing how to make a certain form also shows you how to avoid to make the others. In form ranking, you can list the properties, you can have a nice table, certain values there, but it is important to look at your result as well. You can list your stability, see what competitive slurries do, you can have a look at the melting event, and different ranking criteria can be put on. In the end, it has to be producible. For all ranking is based, can we actually obtain a scalable method in a suitable process solvent? Having a ranking does not necessarily give you the best results. So, when you look to the left, the microscopy picture here shows only a few transparent impurities. These were not detected by any of the other analytics and only visible by microscopy. So don't use only the numbers that you get from your equipment, but also have a look for yourself what actually is the form you're making. We had to do some recrystallization of this compound, and as a result, seen to the right, the crystals grown longer. These are much larger than obtained when the impurity was present. In this case, an additional micronization step was required to get a good material that could be formulated. Finally, two examples. If a stable form, a good solid, was selected. That's the case here in which a free base showed only a poor solubility. We did a salt study and there were several candidates. From that we had a good stable, non-hygroscopic and very soluble salt form. Based on solid state properties, a very good material. Production, a very good material. We did stability tests, diverse hygroscopicity, competitive slurry, and we looked at dissolution in water, and in the end this form was selected. In the tox studies, we decided to go with a freshly prepared injectable, and these studies failed. There were issues that solids were formed when injected. So additional tests were done and we checked what was actually the case. We knew that it was an injection in blood, and the blood pH shows that this salt was not stable. The free base was formed and it crystallizes out. We should have gotten a different selection of the salt if we had knew that the formulation would have been an injectable. So selection of your form is also depending on the intended formulation, the route and the dosage. Second example of did we do the right selection? In this case we had a free acid, only very poor viscous properties. Salt study was done, there were several candidates, and in the end a sodium salt was obtained. Being a sodium salt and the preferred one to take, that was tested, but it shows to be extremely hygroscopic, so it was not selected as the preferred candidate. Second in line, a good stable, non-hygroscopic and good soluble salt form was selected, and actually it gives good results in all tests. The report was shared with the formulation company. That company started to work with this material. And in the end, we had a drug product that failed the stability because it was extremely hygroscopic. The question we got was, did you get the right studies? Did you do the hygroscopicity study on this material as well? We knew that this salt was not hygroscopic, not at all. We had a look at the formulation, and there we saw that one of the excipients was a sodium salt. Making the formulation allowed to change the salt form into the sodium salt, which was extremely hygroscopic. So again, knowing what actually your formulation will be would have avoided this one. But also, if you share not only the successes, but show what was actually a failed experiment, that is also a result, and indicate the reason why a certain salt or a certain form is not selected, that could already avoid possible issues and repetition of certain experiments. Solid state is continuous troubleshooting, trying to reduce the risk as much as possible. So having a broad overview at a very early stage of the development will give you understanding and might give you an avoidance of changes of form in a later development stage. It is very important to have all data, the decisions that were made, the conclusions that were made, accessible for the whole team, and not just a written report with the successes, because the conclusion will be read and nothing else in there. The use of automation that will lead to doing experiments in a smart way, to get the overview to select optimal conditions. It does not give you the final answer. Further, development of the form, checking if it needs to be optimized is required. It allows you to screen different factors, gives you a good diversity in the methods, the solvents, concentrations, as well as the starting materials. That will give you a good overview of the different forms that can be obtained. And thank you for your attention.
Thank you, Dr. Aret, for that excellent presentation. We do have some questions from the attendees. And the first question is, how do you translate cooling rate from a 96 well plate to a reactor for bulk preparations?
Okay, thank you. Let's say if you just translate it as such, saying, oh, this is the rate, that will fail, of course. So there are many ways how to do that. The first one is actually stop heating. You have the linear controlled cooling, and you have the one where you try to keep the supersaturation as equal as possible. So you need to have that translation in what is possible at that large scale, take into account how fast can you cool, as well as how will be the temperature profile across your reactor. So the size of your reactor, that is also one of the things that is important. So there are several steps in between where you need to test which cooling rate is actually suitable for this crystallization, and it is not just a simple translate the exact, let's say, the well plate screen towards the next scale, towards production.
The next question is, crystal size controls such as average size and size distribution are critical to performance and are regulated for APIs. What approaches does Mercachem take to control size?
Yes, so normally controlled crystallization gives you a reasonable, narrow particle size distribution. If not, you can either fine-tune your conditions there, or if that doesn't work, you need to go to a micronization step. You rather want to avoid that part. So the alternatives of looking at different, let's say, seeding methods are also possible there.
Is nucleation and growth of crystals a function of temperature and concentration only, or are other factors such as light and pressure involved?
Yes, of course. Well, I only had this 45 minutes, so I kept it as simple as possible. There are many things that could influence actually the nucleation. That can be, let's say, a local concentration there, changed by even sound. Anything can be actually causing that nucleation. But let's say in the end, you need to have something that starts the nucleation, and that can be a temperature, that can be a seed, that can be an anti-solvent that just enters your solution there. And there are many crystallizations that we still don't understand how they are actually controlled.
What can you tell us about co-crystalline APIs and their future? Does Mercachem conduct co-crystal screening?
Yes, that's a simple answer. We do perform co-crystal screening, but not as the initial starting point. At first, we have a look, how does it crystallize in different polymorphs? Next, can we make a salt? And if not, can we make any coordination there? Working with co-crystals is asking more about your stability study and as well as what is your dissolution behavior. So it requires some more steps before it can go into a manufacturing process. And I must say that it is quite a challenge to have a co-crystal produced at larger scale than just in the lab.
What are your thoughts on the commercial potential of polymorph screening via the application of applied magnetic fields during crystallization?
It is something I did about 15 years ago. At that time, it was something, let's say, not yet very much controlled. I don't know if it will be a commercial feasible process, because you need quite high fields to have some control about it. Could be done just to make some seeds and make sure you have that form as a starting point and see if you can get a normal crystallization from that. It is more of a research field than towards the really production at this scale.
What about surface science technologies that reduce the energetic barriers to nucleation? These can impact nucleation as well.
Yes, these are not tested in our labs at this moment. So it's more like if a certain material does not want to crystallize, it just oils out or anything else is there that avoids a good seed formation there, then we can go to that kind of technologies, but that's more in the field of university, where we can find some more in-depth knowledge about that certain form. It's not something we do in, let's say, a regular screening project here.
Which polymorph to pick for enantiotropic polymorphs? The one stable at ambient temperature or the one stable at high temperature?
Okay, that's, well, there will be, again, not one answer for that. At first, how is it being produced? So if you produce your crystals at high temperature and you can keep that one stable enough just at room temperature, So where is your transition temperature? That is depending on this choice. If it is a high transition temperature and it's easily transferred one into the other, I would go for the one that is at ambient temperature. It requires just a different way how to make the crystals. So the control of that one is very important, that it is not accidentally by cooling crystallization you make already some of the other form and you end up with a mixture.
Theoretically, with different conditions, one compound can make an unlimited number of solid and crystal forms, right?
Yes, unlimited. Indeed, that's theoretically. It has to be stable long enough to detect them. So indeed, you can make an unlimited number of forms. How stable they are, and if you can keep them long enough to analyze, that is the question there. But there is no limit to that.
Why not use DSC with sealed crucibles for controlled cooling as a complementary technique?
We test let's say, crystallization from the melt, as well as see how they transfer from one form into the other by heating just below the melting point. That is something we do on material as received. We see if the amorphous form wants to crystallize once it has had a higher temperature for some time. So indeed this is done, and depending on the material, how it wants to crystallize from the melt or recrystallize at higher temperatures, then it will be something to look at. It is done, it is not done in a screening fashion.
Why is solid form selection needed, as there's only one most stable crystalline form?
Indeed, there is one crystalline form which is the most stable one, but by just doing experiments, you don't know which one it is. It is, and you assume the one you get, let's say, after a few crystallization is that form to go for, but it is not really considered to be the one that is really the most stable one. So you are looking for different forms and looking for conversions into a more stable one or a more suitable one to go for. So the crystallization based on experiments is just to try and hope you find the right one. If you have a diverse set, you will find a good overview. If you combine it with some, let's say, crystallization prediction, you can already see which ones are calculated to be the most stable one. The combination of the two, that will give you more certainty. The accessibility has to be found by only doing screening experiments.
How does the results of a high throughput well plate screen relate to the product at manufacturing scale?
Okay, so when looking at how to make it, indeed there has to be several steps in between. But when you have the form, the crystalline form, that one is normally good reproducible at larger scale as well, if you go from the well plate to grand scale to production. When you look at the particle behavior, so particle size and particle shape, that one is actually not really in relation when you see it in a small scale experiment towards what has happening in larger scale production. So it is something that has to be stepwise, and then you see what you need to control. Is it purity? Is it the particle size? Is it the particle shape? And that's learning on the scale-ups as they appear.
Polymorph prediction software has developed greatly in the past years. Could this replace the many screening experiments and selection process?
No, it will not replace it. It's additional to that one. So the advantage of a prediction is you know which one is actually the most stable one. But the disadvantage there is you need to make it, so you don't know how to produce that form. On the other hand, screening, it is possible that there is a more stable form there that you didn't access yet by the normal ways of the screening that you do. So the combination of the two, that will be the most strong conclusive form to take.
All right, that's all the time we have. Thank you again, Dr. Edwin Aret, for your fascinating presentation, and thank you participants for being a great audience. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you On24 for technology and production services, and thank you Mercachem Syncom for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Kelly McSweeney. Goodbye.